Good afternoon and welcome to the webinar on Infectious Diseases and Biotechnology organized by Spectrum Institute of Science and Technology in collaboration with Edinburgh Napier University. This session will provide you with insights on infectious diseases and the role of biotechnological applications in the diagnosis process. Before we start, I kindly request you to switch off your videos and mute your mics throughout the sessions. And the participants can post their questions on the chat and the panel will address the questions during the Q&A session. Thank you for your cooperation. So the first topic of discussion is on the basic epidemiology of infectious diseases, and the session will be conducted by Dr. Donald Morrison, who is visiting us all the way from Edinburgh Napier University, and he is a lecturer in microbiology at Edinburgh Napier University and also the ENU module leader for medical microbiology at SIST. Dr. Donald, it's over to you. So you can share your screen.
Good morning, everyone.
Good morning.
I am sharing just getting my screen up for you.
Yes, sure.
Okay, are we on?
All right, yes.
Davina, we're on?
Yes.
Okay, good morning, everyone that's listening. It's very different speaking to you from my bedroom, which is my office classroom for the last few months. I was out in Sri Lanka visiting Spectrum at the end of January, and I usually come out every year, every second year, and always enjoy my trips to Sri Lanka. So just a welcome from me and from Edinburgh University, who I represent. Okay, so today I'm going to spend the next 20 minutes telling you about epidemiology. So hopefully by the end of the 20 minutes, you'll be a little wiser about what epidemiology means in terms of infectious diseases. Okay, so if I was going to show you this picture, would it mean anything to you? If I was going to show you this picture, would it make the story any clearer? Where is this place? Why has this man got a mask on? Of course, I'm talking to you about the new SARS-like virus, as it was called back then. So this information was from early in January. So a new SARS-like virus, a pneumonia outbreak in a city called Wuhan, and nothing else was known about it. So this is where epidemiology begins, and where the person who does epidemiology begins his work, the epidemiologist. He finds a situation like this. The authorities have closed down this market, but there was a virus, there was pneumonia, nobody knew anything about it. All they knew is that this city, Wuhan, this market had been associated with people ill with pneumonia. They didn't know what virus it was at one stage. A few weeks before this, the beginning of January, they didn't even know it was a virus. And that's where, as I've said, epidemiology begins. And of course, now we know that the name of this pneumonia is COVID-19. So, my talk is about epidemiology of infectious diseases. And I'm really going to talk to you about what this man who does epidemiology, what is his work? He's called an epidemiologist, and the kind of other way of thinking about him is that he is a disease detective. And hence I've got a picture of our own Sherlock Holmes here asking questions. Who, why, what, when. So that's what the epidemiologists did when they had this pneumonia in a market, a wet market. What was it? Who did it? How did it happen? How did it get in? these are the questions that they ask. This man here, not the man on the right-hand side, but the man on the middle or on the left-hand side, the smaller man, is Dr. Anthony Fauci. He is the director of National Institute of Allergy and Infectious Disease in America and has been there a long, long time. He is an epidemiologist and he's been looking at this disease. Previous to this, he was looking at HIV. So the questions an epidemiologist asked are three. The first one he asks is, where did this come from? Now, the second one we'll deal with later about who did it, and then how on earth did it get into this market, did it get into this place, did it get into this crime scene? Who did it? So, one answer to this question about where it came from was this building here. And this building is the Wuhan specialist virus laboratory in China. So it's in the city of Wuhan, not near the market, but it's in the same city. So some immediately said, this is where the virus came from this laboratory, escaped these either careless scientists or deliberate escape from this lab. And since that, there's been lots of this stuff going on. Our own BBC had this headline, COVID-19 and fake news. There is a lot of it about. And I was wanting to make a poll, get you all to answer a question of whether you actually think that COVID-19 is a hoax. Is it fake news or is it not? So I was wanting to do that survey, but we don't have any opportunity to do it on Zoom. The survey was done in the United Kingdom, and more than a fifth of people in England believe that COVID-19 is a hoax. That's over 20% of the people in England, and I'm astounded at that. As a scientist, I'm astounded. And as a scientist, I am frustrated by fake news about COVID, about vaccines. And what we need to do as scientists is to be able to show where the fake news is going wrong in its assumptions. And one way to do it is by using studies. So this was a study done in April 2020, published in the major journal Nature Medicine, and they looked at the genome of this virus. And by this stage, there have been over 20 viruses sequenced. They looked at these sequences and they asked, is this a laboratory constructed virus? Because this was the fake news, and it come from this lab. They looked at it, they looked at the sequence, and usually when you construct a virus in laboratories, and this can be done, nobody's denying that, but usually you begin with a scaffold of another virus and you add your toxigenic or infectious one on top of it. This one was a natural virus all the way through. And the conclusion they had was that our analysis clearly shows that SARS-CoV-2 is not a laboratory construct. It was not purposely manipulated virus. So for me, this is the kind of stuff that people need to hear. The science community need to be telling this. The other one was 5G. So 5G is responsible for the mutations for coronavirus. Lots of these fake news. I would direct you to this website, Snopes. You can just search on anything that you're afraid of that may be fake news, and they will give you some solid information about it. Okay, so the fake news, by showing this map of the cases, daily confirmed cases of COVID, to me, this itself is enough to do away with this fake news, this COVID being a hoax. If it is, then there's many, many countries involved in this hoax, and that is very difficult to believe. So this is a map you probably all have seen. At the top, you will see United States, Brazil, India, with the largest number of cases, the largest number of population, so may not be unexpected. However, China, which is the largest country, has not got these high cases. So the cases for China were much lower. They managed to control it. So that's one thing. The other thing you can see from this is that the United Kingdom, not the largest country in the world or anything like it, is a third, fourth in line here. Yourselves, Sri Lanka, down at the bottom. You've had a bit of a blip, maybe in March, but still below the 100 cases. So Sri Lanka have done very well in terms of containing the COVID virus. In terms of cases worldwide, we are now at 10.2 million. United States, 2.5 million. Brazil next, Russia, India, and the United Kingdom have got the infamy of being fifth in terms of the total number of cases. We have not done a good job in containing this virus. Deaths, of course, the thing that's most important or most impacts us most. Again, similar chart, similar people at the top, Brazil, United States, India, and unfortunately, again, the United Kingdom. You have again done very well here. So your health services, the people in Sri Lanka have complied well with the lockdown restrictions. Only 11 cases in the whole of this time. Worldwide, 0.5 million. United States, 128,000. Brazil next. And us here in the United Kingdom, fourth in line, 43,000 deaths. If we take nursing homes in, it was 48,000. So we have not done well here. And in the next few months, I'm sure the reasons for that will come out. This is a chart for the United Kingdom, and we went up high in April, May, and then began to come down. And now we are easing lockdown, although there's a fear that at the end of this chart, we are leveling off, which is not what we would like. If you do level off, it means that the virus is not declining and lockdown may have to resume. In fact, one city in England, Leicester, has imposed lockdown again because of this problem. Anyway, this is me trying to show you that this is not a hoax. This is a major problem. So back to the topic. I was talking about epidemiology and this disease detective. The first thing he did was he asked where was it from? If it wasn't from the lab in Wuhan, where did it come from? The virus itself was discovered after they sequenced it, cultured in tissues, and it was found to be coronavirus. It is named because of these crown-like spikes that it contains. But where from? An animal reservoir is agreed by everyone who is working on the sides of this. And interestingly, coronavirus is found in many, many animals. This is not an uncommon virus. Pigs, camels, bats, cats, and there are hundreds of different coronaviruses. This is not an uncommon one. But in terms of humans, there are only 7 that infect humans. 4 of them are just responsible for the common cold. And when you get the common cold, once a year, twice a year, more if you're unlucky, you are very likely getting one of these coronaviruses that's causing it. Of common colds, 50% are caused by rhinovirus, but the other 50% are caused by 8 or 9 different viruses, 4 of them being the coronavirus. However, there have been 3 coronaviruses in the last 20 years that have caused serious disease in humans. As you notice, they are all beta coronaviruses, similar to the OC43 that we have as a common cold. SARS in Hong Kong and China, again, it began 2002. There was a big pandemic lasting for 2 years, and then it disappeared. So that's kind of good news, possible good news in terms of what we are facing today. SARS-CoV-1, as it was called, it wasn't called 1, but now we've got 2. It's very closely related to the one we have now. Then in 2012, in the Middle East, MERS, Middle East Respiratory Syndrome, another beta coronavirus, 2012, it has not disappeared, but it has remained within Saudi Arabia and some of the surrounding regions. One transferred to South Korea, which was then curtailed, but it is still around. From camels, drinking camel's milk, being near camels. And then, of course, the one that came from Wuhan, or began in Wuhan, at least initially, that was where we found it, SARS-CoV-2. COVID-19 is actually the name of the disease. SARS-CoV-2 is the virus. 2019, it's still with us. How long? Will it be like SARS-CoV-1? Or will it be like MERS? Or will it be like OC43, which began as a pandemic and now is the common cold? And this is a story of OC43, a possible route for our COVID-19. 1890 it was found, again a spillover from animals to humans. Cows to humans, cows got it from mice. It caused a major pandemic in 1890s. Killed 1 million people worldwide. Previously, it was thought to be flu, but some genetic analysis found there was actually this OC43 coronavirus. Today, circulates widely, doesn't cause us much problem, sniffly nose, feeling miserable for a couple of days. Is this the way that COVID-19 or SARS-2 is going to go? We don't know yet. So we've asked, where is it from? We've asked, who did it? Now the third question that the disease detective asks is, how did it get in? How did it cause this crime? How did it get into the house? How did it get into us? Where did it come from? And this is about transmission. In terms of epidemiology, there are 3 major routes by which you get a disease. And if you want to learn about infectious diseases, this is a very good way to learn to approach it. If there's a disease, ask, how does it move? Where does it come from? And how does it get in? And there are 3 basic ways that a infectious disease can move. Person to person. And there are 2 subdivisions. One is airborne, and one is by direct contact or intimate contact, like sexually transmitted diseases. And of course, we know that COVID-19 or SARS-2, this is the way it moves. It gets in through person-to-person spread. Is it airborne or is it direct contact? We know it's not intimate contact, but it's airborne direct contact. It's between these 2. The other 2 ways that you can get an infectious disease is by some vehicle. So the 2 main ones are via water, cholera being a prime sample of that, or food. And again, many examples of that, many viruses many bacterial infections, salmonella being one in particular from food. The third way you get an infectious disease is through vectors. So something carries it to you. It can be an animal, and the plague, Yersinia pestis, which causes the plague, was carried by the rat, which carried a flea, which carried the Yersinia pestis, the bacteria. Arthropods, of course, the most famous will be, or infamous, is malaria, carried by, or the plasmodium carried by Anopheles mosquito. So 3 ways we're looking at COVID. Is it airborne spread? If it is, this is the model that you're looking at. Let's look at this, first of all, beginning from the closest. So at the bottom here, you've got 1.5 meters. This is where droplets, and these are red, big red is big droplets. So when you breathe out, or when you talk, or cough, or sneeze, you are putting out droplets of moisture, and inside these droplets, you will have whatever is in your throat, or in your nose, or in your mouth. If you have SARS-2, then this will be inside the droplet. So these red ones are large droplets of water, moisture. If you're talking, then, and you have SARS, then your droplets will drop around 1.5 meters away from you, because they're quite heavy and they get dragged down. However, if you cough and you have SARS-2, then obviously the cough, as we know, has got a certain strength. This will take these droplets, large droplets, to about 2 meters. So if you have somebody who has SARS, has COVID-19, you want to be at least 2 meters away if he's coughing. However, if he sneezes, higher velocity, you want to be a long way away from him, at least 6 meters, because his droplets, these large droplets, will not fall to the ground until at least 6 meters. But this is okay, because people who are sneezing and coughing, you know that they're ill, and they will actually be quarantined themselves, or you will be aware of it and you will stay away. The problem is people who are just talking to you may be asymptomatic, and they will be talking and releasing the viruses, and we know that for 2 days, somebody who is asymptomatic is shedding the virus. And this is the problem. This is why quarantine, 2 meters, wearing masks is so important. However, like everything in life, in biology and science, it's not that simple. We have these little dots here, which are not droplets, they are aerosols. These are small, they are droplets of moisture coming out of your mouth, but they are very small. And these can also carry the virus, and unfortunately these will stay airborne for a long time, several hours, and they will travel. And this is kind of the extra issue in terms of spread. So in terms of spread, you've got the 3 ways. You've got person-to-person direct contact within 6 feet. You will get the droplets going direct, droplet to droplet, the red one. You will get fomites, where the droplets go into some surface and then somebody touches it. This is indirect contact. Or the third one, the more dangerous one, aerosols. They will be up here in the air and they will stay in the air for some time. So the last thing I want to tell you about before I finish is the severity of COVID-19. Now, some hoax, fake news will tell you it's just flu. Well, sometimes it is just flu, but not all the time. And about 10% of people are getting severe disease. And if you compare it with the common cold, OC43 for instance, which will give you the common cold. An interesting observation has been that coronavirus OC43 infects the nose, the throat, and the eyes, through the eyes. If you look at another of these serious ones, coronaviruses, the MERS, Middle East syndrome, it affects the lungs and only can infect the lung cells. It doesn't infect your nose and your mouth. However, SARS-2 is different. It can infect cells, not only in your nose and mouth, but also in your lungs. And this is one of the reasons that SARS-2 is more severe than any of the previous ones. It can attach to any of these surfaces, get access, and also cause a more serious disease in the lungs. Okay, thank you for listening. That's the disease detective working on the new SARS-2. Thank you.
Thank you, Dr. Donald. So moving on to the next session, which is on the topic of viral infections and immunology in dengue especially. And I invite Professor Peter Barlow, the Professor of Immunology and Infection and the Head of Research at School of Applied Life Sciences in Edinburgh Napier University to conduct the session. Over to you, sir. So you can share your screen now.
Good morning, everybody.
Good morning.
Let me just share my screen. Oh, okay, sorry, hang on. I just need to enable this in my system preferences. Oh, sorry, I can't share my screen until I quit and rejoin. Just give me 1 minute.
Yes, sure, sir. Participants can post any questions on the chat, which will be discussed at the end of the session.
Hello, I'm back. Right, sorry, I'll try this again. Oh, it says that the host has disabled screen sharing.
Yeah, we'll sort it out and get back to you soon.
Thank you.
I think you can share now.
Yeah, I think I
Professor.
can do it now.
Yeah.
Fantastic.
All right. Great.
Okay, so can you hear me okay?
Yes, we can hear you.
Fantastic. Okay, well, thanks very much for the invitation to speak. And Donald, thank you so much for such an elegant talk. So I'm going to talk to you today about new antiviral therapeutics that we're doing some work on in the School of Applied Sciences. And they're known as host defense peptides. And they're a key part of the innate immune system, but they have incredibly potent antiviral activity. So I'm just going to talk about a little bit of the work that we've been doing with these host defense peptides and some different viral infections. So host defense peptides are incredibly exciting next generation antimicrobials. I mentioned that they're a key part of the innate immune response against infection, and they're actually found in most organisms, including animals, insects, plants, and humans. They are very tiny molecules. They're normally only about 12 to 50 amino acids long, so they're very, very tiny. And in humans, we've got two main families, the cathelicidins and the defensins. And what happens is when you get an infection or inflammation, these host defense peptides are upregulated and released by your immune cells and can destroy the invading infection. And the reason that they can do this is because they have a cationic charge. And what that means is that the peptides are incredibly efficient at binding to biological membranes. So here's a nice model which shows these tiny peptides binding to the surface of a biological membrane, and then they can actually create pores very, very rapidly in that biological membrane. So they act a lot like a detergent. They actually punch holes in the membranes of pathogens, which is why they're so efficient at killing bacteria and destroying viruses. So I work on the only human cathelicidin. It's called LL-37. It's called LL-37 because its sequence starts with 2 lysine residues, and it's 37 amino acids in length. And this is what it actually looks like when you do a 3D model of it. You'll notice it looks like a coiled spring. So LL-37 is actually stored predominantly in your neutrophils. So all the circulating neutrophils in your body are absolutely packed full of LL-37. And it's stored as an inactive peptide. So essentially inside the granules of the neutrophils, you have the pro-peptide, which is called HCAP18. And when you get infection or inflammation, the neutrophils are recruited to that site of infection. The neutrophils degranulate, they release all their contents into that area. And the enzymes that are present on the surface of the neutrophils will actually cleave the peptide and make it become active. And you can find the peptide in most fluids that are found in the body, like lung fluid, plasma, sweat, saliva, tears. It's pretty much found everywhere in the body. It can also be released by lots of different cell types in the body, and these include epithelial cells, mast cells, and macrophages. So it's not just the neutrophils that produce them. If you want to force cells to produce LL-37, you can actually treat them with compounds such as vitamin D or butyrate, which is a short chain fatty acid, and that actually induces the cells to produce lots of LL-37. Interestingly, there are people that have chronically elevated concentrations of LL-37 in their bodies. These include people that are suffering from cystic fibrosis, lung disease, and people with psoriasis, the skin condition psoriasis, actually have huge concentrations. There we go. Can you hear me okay? Just said I was muted there.
Yes, I can hear you.
Oh, sorry, it just came up on my screen as being muted. LL-37 actually has a huge range of antiviral activities. Okay, so there's you can find cathelicidins from lots of different species that they have massive antiviral effects against a range of different viruses. And defensins, the other human family of host defense peptides, also have antiviral effects. And at the moment, we know that the LL-37 has antiviral effects against viruses such as vaccinia, RSV, HIV. We know it's antiviral against dengue. And we were the first to discover that it was antiviral against influenza and rhinovirus, which Dr. Morrison very kindly mentioned earlier as one of the main causes of the common cold. What we don't know as yet is whether LL-37 has antiviral effects against coronavirus. but we have 2 grant applications which are awaiting judgment, so hopefully we will be able to answer that question very, very soon. So I'll just talk for a couple of minutes about rhinovirus and influenza, because that's where we started the work. Rhinovirus and influenza are 2 of the most common viruses that are associated with viral pneumonia. And viral pneumonia as a condition, is a particularly prevalent global problem. So the World Health Organization thinks that there's around 450 million cases of pneumonia each year, and around 4 million people die from that. So because rhinovirus and influenza are so closely associated with viral pneumonia, we wanted to see what the effects of host defense peptides against rhinovirus and influenza were. Now, rhinovirus, as Dr. Morrison mentioned, is a relatively innocuous virus, but actually it has been shown that there's a relationship between rhinovirus and bronchiolitis in infants. And bronchiolitis is an acute inflammatory condition in infants, which can be life-threatening in some instances. But also, it's been shown that rhinovirus infection can also place children at an increased risk of developing asthma in later life. If you're an adult and you have a pre-existing lung condition like asthma or chronic obstructive pulmonary disorder, and you catch a rhinovirus infection, it can actually be quite life-threatening. So healthy people will contract rhinovirus anywhere between 2 to 4 times a year. If you have a child, you will likely contract it a lot more. And unfortunately, it just at the moment doesn't have an effective treatment or an effective vaccine. So antiviral drugs against rhinovirus as well as influenza are desperately needed. This is what rhinovirus actually looks like. The image on the left was taken by a PhD student of mine, who's now a postdoc, Dr. Philippa Henderson-Souza, and that's a scanning electron, a transmission electron micrograph of rhinovirus, which we took down at the University of Oxford. So we did a series of very simple experiments where we took LL-37 and we exposed the virus to LL-37 and looked to see how much virus was actually remaining, whether LL-37 could actually have antiviral effects against rhinovirus. And we took a few different treatment approaches. I've just highlighted 2 here. So in the graph on the left, we treated the virus with peptide for about 1 hour and then allowed the remaining virus to infect cells so that we could quantify how much virus was remaining. The graph on the right, we treated cells with rhinovirus and LL-37 at exactly the same time. And what you'll see is, if you look at the black bar on the very left of both of those charts, that's just virus itself. Obviously, with no treatment, there's 100% of virus remaining. But when we treated with LL-37 at a range of concentrations, you can see that in some instances there was around a 95% drop in the amount of virus remaining. Now that's a huge drop. That's an enormously potent effect. Before I came to Edinburgh Napier, I worked for the US Centers for Disease Control and Prevention. And there we did a mouse study where we infected mice with a lethal dose of influenza. And we treated those mice with either a control, which is in the black bar. We treated them with zanamivir, which is a common anti-influenza drug. And we also treated them with either LL-37 or scrambled LL-37. And scrambled LL-37 acted like a control. It had all the same amino acids as LL-37, but scrambled up into a random sequence. And what you see there, if you look at charts A and B, is that LL-37 had almost identical efficacy to zanamivir, which is the commonly used anti-influenza drug. And that tells us that LL-37, in mice at least, has incredibly potent in vivo effects. You can see that without treatment, all of the mice died, but with zanamivir and LL-37 treatment, around 75% of the mice survived. And remember, this is a lethal influenza infection of mice. If you look down at the bottom there, chart C, that shows the amount of virus that was actually remaining in the mice after treatment with LL-37. And what you do see is you see about a tenfold drop in the amount of virus when you treat the mice with zanamivir or LL-37. So that's quite a substantial drop in virus, but it maybe doesn't explain why the mice survived. So there might be other things happening here. We also treated the mice with cathelicidins from other species. So as well as the human LL-37, we treated the virus and the mice with cathelicidins from the pig. So in the center top there, protegrin 1, and we found that protegrin 1, which is a pig cathelicidin, was just as effective as LL-37 in terms of halting the amount of virus replication that was taking place. We treated the virus with MCRAB, which was the mouse version of LL-37, and that also had some significant effects. However, with with influenza, what we found was that protegrin didn't have any effects. So in contrast to rhinovirus, where protegrin was incredibly effective, protegrin could not rescue the mice that were infected with influenza. So what that tells us is the human cathelicidin, LL-37, has antiviral effects against flu, rhinovirus, but the pig cathelicidin, protegrin, only has antiviral effects against rhinovirus and not against flu. Interesting, we look to see whether cells could actually upregulate LL-37 during rhinovirus infection. And what we found was that yes, actually, when you get a rhinovirus infection, your body actually upregulates production of LL-37. So that tells us that LL-37 is a key part of the innate immune response against rhinovirus. Now, you'll have heard a lot about inflammation recently, mainly in the context of SARS-CoV-2. Inflammation can be incredibly beneficial. Inflammation is required to clear a viral infection. And in infections like rhinovirus and influenza, inflammation can help to clear the infection. However, what can happen is if the inflammation is poorly controlled or it is excessive, then actually that can be quite a damaging process. And it's actually associated with clinical severity in terms of rhinovirus infection. The reason I mention it now is because SARS-CoV-2 or COVID-19 has been shown to cause inflammation throughout the body, and recent developments have shown that the steroid dexamethasone can be beneficial in reducing inflammation. So actually, we think it's quite interesting to see whether peptides like LL-37 can reduce inflammation. Oops. What we did was we treated cells with rhinovirus, and then we exposed those cells to LL-37, and we looked to see whether LL-37 altered the inflammation that's associated with viral infection. So we found that when we treated cells with human rhinovirus by itself, that's the black bar, that a lot of inflammatory cytokines were upregulated. So things like IL-8, IL-6, IL-1 beta. But when you treated those cells with LL-37, we found that the levels of inflammatory cytokines, the concentrations, the gene expression of the inflammatory cytokines actually decreased. And what that told us was that LL-37 could actually be beneficial in reducing the amount of inflammation that's associated with viral infection. Now, that can be a good thing or a bad thing, depending on what stage the infection's at, or what the inflammatory status of the patient is. But we believe that this merits further investigation in terms of a treatment for viral infections where inflammation can be quite a damaging process. We also tested this observation in mice with influenza, and we found exactly the same things. That when mice were treated with or were infected with a lethal dose of influenza, the PR8 only treatment and all those charts there, that the concentrations of inflammatory cytokines substantially increased. But when the mice were treated with PR8 and LL-37, the LL-37 was able to ameliorate or reduce the inflammatory burden in those mice. So that might help to explain why a greater proportion of mice survived when they were treated with LL-37. So what's our future and current work at the moment? Well, what we're doing at the moment is we know that LL-37 has antiviral effects, and I collaborate with the CDC still in terms of developing new peptides that could potentially be used to treat these infectious diseases. And what we're doing is we're developing variations of LL-37. So LL-37 that has different amino acids inserted here and there to make it more effective. And we're looking to see whether we can generate almost a super LL-37 that could be used to treat viral infections. We always get a lot of media coverage for the work that we do because people hail it as a cure to the common cold, but that's not our aim. We're not trying to cure the common cold. We're trying to develop antiviral therapeutics for rhinovirus. I just want to mention our current project, which is funded by the Medical Research Council, as well as Ristekdikti, which is the Indonesian Ministry for Science. We're looking at host defense peptides as antiviral therapeutics for dengue virus infection. And this is a 3-year project that's being conducted in collaboration with investigators from the Eichmann Institute for Molecular Biology in Jakarta and University of Indonesia in Jakarta as well. And I'm sure that dengue virus is a pathogen that you guys in Sri Lanka are all very familiar with because it has a huge global burden of disease. So I've been lucky enough to go out and work with my partners in Indonesia. And as you can see, I've been treated to some wonderful food. And I've been lucky enough to have those guys visit our facilities at Edinburgh Napier. And we're engaging in this fantastic collaborative project, looking to see whether we can make host defense peptides as antiviral drugs for treating dengue infection. And we're still at relatively early stages. There is existing evidence in the literature. So this was a paper from 2017, which showed that if you treat dengue virus with LL-37, that it does have antiviral effects at relatively low concentrations. The interesting thing about this paper is that they also showed that if you infect cells for 24 hours with dengue virus and then treat with the peptide, that the peptide still had antiviral effects at this stage. This is some of the recent work that we've done, looking at whether host defense peptides are produced in response to dengue virus infection. So we took blood cells from healthy donors, and we exposed those blood cells to 2 strains of dengue and measured the expression of cathelicidins. And interestingly, what we're starting to see is that cathelicidin expression appears to be decreased in response to dengue virus infection at later times. Now, this is quite interesting. This is very early stage work, but this suggests to us that if dengue virus has the potential to decrease the amount of host defense peptides that are being released, that there could be benefit in treatments that would boost the antiviral response to dengue. So what we're doing in this study is we're looking at patient samples from acute and severe dengue patients, and we're measuring the amount of cathelicidins and vitamin D that they have in their serum. We're also looking at whether cathelicidins, like LL-37, can modulate cell death pathways in cells that are infected with dengue, and we're looking to see whether vitamin D, LL-37, and butyrate can be used as therapeutics for dengue infection, because both vitamin D and butyrate can upregulate LL-37. So perhaps it can help to switch the balance. We're also trying to work on creating a drug delivery platform for our host defense peptides. I didn't have time to go into it in detail just now, but host defense peptides are actually susceptible to degradation if you just deliver them in their native form. So what we're trying to do is develop a nanotechnology drug delivery platform for our host defense peptides. And as I mentioned, we have a couple of grant applications in at the moment to look at the effects of LL-37 against SARS-CoV-2, or the novel coronavirus, because we believe that LL-37 will also be able to act against this pathogen. So I'd just like to finish by acknowledging all the people that actually did the work, all of my collaborators, the guys in Indonesia, as well as Jan Pol from CDC, the students that have undertaken projects in my lab, as well as the funders, including the MRC, RISTEC, the Newton Fund, Chief Scientist Office in Tenvis, Scotland. So thanks very much for your attention.
Thank you, sir, for that very interesting findings and all the information shared. So after that session, we move on to the last session of the day before we get into the Q&A session. So again, a kind reminder for all the participants to share your questions with everyone so that everyone will see your questions. Okay, so the last session will be conducted by Mr. Channa Tharanga, the Senior Lecturer in Immunology and Molecular Genetics and the Head of Academics of Spectrum Institute of Science and Technology, and also a founder member of the Human Genetics Society of Sri Lanka. And he will be conducting the session on genetic diagnostics of COVID-19. Over to you, sir.
Hi, everyone. I think you all can hear me clearly. So, hi, Donna.
Yes.
Thank you for your contribution. So we are moving into genetic diagnostics of COVID-19. So, give me a minute. Sorry. Okay, so we will be discussing about the introduction, then the principle of RRT-PCR, methodology and how to interpret COVID-19 test results, limitation of this, and also the conclusion. So now the thing is, we always, even in Sri Lanka, we always hear, like, you know, to increase the number of PCR tests, then RT-PCR, and then some people suggest, why can't we increase it to 7,000 PCR per day, 8,000 PCR per day, how they are doing the PCR. So there are a lot of questions. Actually, you know, even though we very simply or lightly mention about the PCR, this is not a very simple technique, especially when it's come to the developing the techniques. It goes hand in hand with bioinformatics. Without the bioinformatics knowledge, we could not have developed these tests. So, and after developing also, when you try to introduce it to the different labs, sometimes, I mean, if you actually have done this PCR, then you guys will know that when you try to even introduce the test to the lab, you have to go for the optimization for each and every lab. Sometimes, whatever the procedures or the protocol works in one lab, sometimes might not work with some other labs. So there are issues. So, however, here today we are looking at the basic principles of this genetic diagnostics of COVID-19. So as Donald mentioned, actually the SARS coronavirus 2 is a positive sense RNA virus. So when we develop any diagnostic methods, especially genetic diagnostic method, this is one of the most important information that we need to look into. This is a positive sense RNA virus because the methods that we are going to apply in order to detect the viruses or any other pathogen can differ from a pathogen to pathogen, especially when it comes to the viruses. It can be a bit complicated because there are so many different types of viruses. Now, this comes under group 4 Baltimore classification. You know, there are DNA viruses, then there are RNA viruses. Even in DNA viruses, there are ssDNA. Then in RNA viruses, you have positive sense RNA viruses. Sometimes you have the negative sense RNA viruses. So likewise, there are so many differences. So based on that, we need to decide on the technique that we are going to use. So this belongs to the Nirovirale order. As previously also mentioned, there are 4 main subgroups: alpha, beta, gamma, and delta. That also based on their genetic structure. And until December 2019, we only knew about 6 of these viruses. Out of that, 4 causes the mild common cold type symptoms in immunocompetent people. Then other 2 have caused pandemics in the past 2 decades, like, you know, SARS and MERS. SARS had about 10% of the mortality rate, and MERS had about 37% of mortality rate. And SARS-CoV-2 or SARS-CoV coronavirus 2, it belongs to the beta coronavirus, SARS coronavirus 2, and it shared about 79.5% similarity to SARS coronavirus 1, which causes the SARS pandemic in 2002-2003 period. So it's somewhat genetically similar to SARS coronavirus 1, which caused the SARS infection. Now, when we look into the principles of diagnostics, the thing is, most of the people, again, most of the students especially, do these mistakes again and again. When you say RT-PCR, someone thinks RT stands for real time. Actually, RT stands for reverse transcription. The simple R in front of it, or simple Q, represent the real time. So this is actually a combination of real time and reverse transcription PCR. So this virus, as I mentioned before, has a positive sense RNA genome. So usually what we do is we need to isolate and purify the RNA from upper and lower respiratory specimens. And then we need, now this is an RNA. So PCR techniques are developed for DNA. So what should we do? Now, if you have an RNA, you should transcribe it to cDNA using reverse transcription. Now, again, when you do this, you can do it in 2 steps. You know, reverse transcription in 1 step, and then the PCR technique in the another step. But sometimes we can combine it, and, you know, as a multiplex, everything together, we can do it. So, whatever the cDNA we transcribe, then we can amplify them using real-time PCR instrument. So once we design this cDNA, we have to design oligonucleotide primers and probes for the detection of this SARS coronavirus 2. Actually, this is when it comes to the developing, this is one of the most time consuming process. So here we have designed probes for the nucleocapsid gene in the coronavirus. And actually, when you do this, the probes that you design, one thing is you have to select a region of this nucleocapsid gene. After you select the region, you have to design the primers. Now, primers, you don't have to be specified only to this version of the coronavirus. It can be a common primer also for some other coronavirus, because it's sometimes a bit hard if you're going to design a very specific primer. But when you design the probe, probe should be very specific. Probe should be very specific for that virus that we are looking. Like, if we are looking at the coronavirus 2, then whatever the probe, the regions where we have designed the probe should be very specific. Only for that virus, not for any other versions, not for even coronavirus 1. Otherwise, you can't detect. Then again, we have to design, we have to use a primer probe set to detect the human RNA P gene. Actually, we use it as a control sample to check actually whether we have done the extraction of RNA properly. Because now sometimes we get the negative results, we can get the negative result if we haven't extracted the RNA properly. So then we have to verify that our negative result is not due to the non-availability of the RNA in our sample. And also we should verify our RNA extraction method for each and every sample that we test. So that is why we are using another prime and probe set to detect the RNase P gene, a region in RNase P gene. So that means if this region, if you can detect this region, that means, yes, even if there is a negative result for coronavirus, if you have positive result for this, that means we have extracted the RNA properly. So okay, so now again what happened here is, now once you start the PCR, the probes goes and anneals to the specific target sequence located between the forward and the reverse primer of our target region. Then during the extension phase of the PCR cycle, there is this 5' to 3' nuclease activity of Taq polymerase. It degrades the probe. When the probe is degraded, the probe is attached to a reporter dye and the quencher dye. So once the extension occurs, the quencher dye and the reporter dye separates from each other, which gives the fluorescent signal. Now, with each cycle, the intensity of the fluorescence will increase. So then this fluorescent intensity is monitored with each PCR cycle using the real-time PCR system. You can see the probe goes and attached to the cDNA or DNA. Then when the primer also binds, then during the extension phase, the cleavage occurs, then the signal can be detected. If probe cannot bind, then you won't be able to detect the signal. Now we have to look into the methodology. Usually we take upper and lower respiratory specimens from the nasal cavity or oropharyngeal swabs, sputum samples. Then and also after obtaining the samples, we have to extract the RNA. When we do the extraction of RNA, we can use HSC or we call it human specimen control, in order to see whether, in order to validate or in order to see whether we are doing the proper extraction process. And it will demonstrate to us that we have successfully recovered the nucleic acid. So we can use human specimen control in order to see the validity or whether we have done the extraction of RNA properly. Then we can prepare the master mix in singleplex or multiplex manner. Different labs use different protocols. If we are using the multiplex, then usually we use 2 primer sets and 2 probes to detect the specific regions of nucleocapsid gene of the novel coronavirus. And also, we will be using 1 primer and probe set to detect human RNA-sp gene as control, internal control. Then when we look at the controls that we use during this, we have to use a negative control without a template, okay? A negative control without a template to eliminate any sample contamination during the assay run. So usually we use nuclease-free water. Then we have to use a positive template in order to determine whether our assay is performing as intended. Like, you know, we can get a positive control sample, but usually the positive control doesn't have this RNA-sp target. And then we have to use an internal control to target the RNase P gene in order to see whether we have extracted RNA properly. Then we also have to use an extraction control to ensure the as a negative control. So, and in the negative control, in the negative extraction control, it is a previously identified negative patient sample. So we are using all these controls in order to validate our results. Now, how are we going to interpret these COVID-19 test results? One thing we need to remember is that no test gives 100% accurate result, and this is a novel test. Like the virus, this is a novel test. So there is no gold standard techniques yet, or none of the techniques have been validated or suggested as gold standard test to determine the COVID-19. However, now this is what you can expect in a multiplex PCR, and this is how you need to report. Now, if you can see, if you detect both SARS-CoV-2 in N1 region and also N2 region, if it is detected, then we report as the patient is positive for the SARS coronavirus 2 or COVID-19. So that means it's detected. But you might, I mean, if you remember, even in Sri Lanka, we had several incidents that first it was reported as positive. Then again, once you do the test, it was reported as negative. Actually, that is because it's indeterminate cases where sometimes only one target is positive. So that means if only one target is positive, sometimes you might have to do the test again. It may be due to some other reasons that you are getting one target is positive. So once you repeat the test, if it is again, if both the targets are positive, no problem. You can report it as detected. Otherwise, you have reported it as not detected. Then, if you get both N1 and N2, the specific regions in the coronavirus genome negative, but RNA-sp region as positive, that means you had RNA in your sample, there was no problem in RNA extraction or anything, and actually you don't have the viral fragments. That is why you got N1 region and N2 region as negative, while the control RNA-PG, which we all have as the positive. So that means SARS-CoV-2 is not detected. Remember, we report it as not detected. Then, if nothing is positive, like even if the RNA-sp region also not positive, that means maybe we haven't extracted our RNA properly. That is why we are getting the negative result. So again, this makes the invalid result. So we have to repeat the sample. Now, when we look at the sensitivity and specificity, this is also sometimes most of the students, they have the problem with these two terms. Actually, sensitivity is the proportion of patients with disease who have a positive test or the true positive rate. True positive rate. Now, if 100 people, if they have the coronavirus, if out of that 100, once you do the test, if only 70% detected as positive, that means the sensitivity of this test is 70%. The specificity means the patient without the disease who have a negative test or true negative rate. Without the disease, reported as negative. Now, if 100 people doesn't have, if they don't have the coronavirus or COVID-19, and if 95% detected as negative and only 5% detected as positive, that means the specificity of the test is about 95%. Now, there are so many reports about this genetic testing, so many reports about the sensitivity and the specificity of this, but we can suggest it's about 70% sensitive and 95% specific. That means even if you are reported as not detected, still there is a chance that actually you are being a COVID-19 patient, even if your test result means it's not detected. Even if the test result does not detect it, you might have the COVID-19 because its sensitivity is around only 70%. This can be changed, I mean, lab to lab and the region to region, but this is the overall sensitivity that they have taken. So, if you see the limitations, now the problem is this diagnostic panel has only been established in the upper and lower respiratory tract. And the other thing is negative results do not preclude COVID-19 infection and should not be used as a sole basis for the treatment. Even though you get the negative results, that doesn't mean that it's completely negative. Then the false negative results may occur if a specimen is improperly collected, transported or handled. And again, the false negative results may occur if the amplification inhibitors are present in the specimen. And also, if the virus mutates, this is very important, if the virus mutates in the target region, we will not be able to detect the coronavirus too. And also, just because you detect the coronavirus, that doesn't mean the causative agent for the clinical symptoms is only the coronavirus too. There can be some other viruses also which do the co-infection. So the conclusion is, usually it is better to carry out at least 2 tests with a time gap, and also even if the negative results are being given out or if it is not detected, better to perform the test twice if the pre-test probabilities are high. That means if you present very similar symptoms, even with the symptoms very similar to the COVID-19, if it's not detected, without concluding that the patient is negative, better to perform another test to confirm the result. So because of these false negative results, the patient can go into the non-COVID-19 wards, or it can lead to the spread of the disease through the hospitals and also in the community. Then all the patients should be counseled about the limitation of the test. And if the patient with a single negative test but strongly suggestive symptoms of COVID-19 should be advised to self-isolate. Okay, so thank you. And if you have any questions, you can post them.
Thank you, sir, for that very informative session. So we have few questions online. I think the first question from Kokul is to Professor Peter Barlow, I guess. Yeah. So Professor, if you could, so given data, the L-137 showing significant reduction in rhinovirus percentage, what exactly impeding in producing antiviral vaccine against rhinovirus using LL-37?
Okay, so that's a really good question. So what we're talking about is using it as a therapeutic, not as a vaccine. So a vaccine's for protection, a therapeutic is for after you've got the infection. The problem with using peptides at the moment is that when they're exposed to enzymes and serum in the body, they can sometimes be inactivated very quickly, which is why we're trying to create versions of LL-37 that do not get inactivated and do not get damaged by natural processes in the body. So yeah, that's an incredibly good question, but it's really why we need to deliver the peptides using some sort of drug delivery platform in order that they are able to cope with the stresses of being in the body, essentially. I hope that makes sense.
Okay, I think there's one more for you. An
Sure.
acceptable inflammation is required in viral inflammation. Taking anti-inflammatory action to account, what is the take in using LL-37 as a prophylactic treatment?
So that's a fabulous question. The main barrier to that is we don't know what will happen if someone takes LL-37 for a long period of time as a prophylactic. The second barrier is that it's incredibly expensive. It can cost several $1,000s for just a few milligrams of purified LL-37, which is why my grant applications are so expensive. But there is an alternative, and that is vitamin D status. So it's been shown that your vitamin D levels are directly correlated to your LL-37 levels. So if you live in a cold climate where you don't get a lot of sunshine or you don't take a lot of vitamin D, then you can be vitamin D deficient. And in respect to that, you can also be cathelicidin deficient. So one of the many benefits of taking vitamin D is that you can increase your cathelicidin, your circulating cathelicidin. Not to a great extent, and there's a lot of complications with that. But in terms of LL-37 as a prophylactic, the easy answer might be just make sure that your vitamin D status is within the healthy normal range.
All right, thank you. And I think we have two questions to Mr. Chandna as well. One is from Nuska. What are the chances of false positives or negatives of COVID PCR, and how are they verified?
Actually, the false positive rates are very less. The false positive rates are very less, but the false negative rates can be a little bit higher, like, because that is why the specificity is about 95%. That means, if you are detected as COVID patient, then there is a less chance that it being a false result. So the false positive results are very less, like about 5% most of the times. But false negative results, as per worldwide reports, the false positive results are higher. Actually, PCR, they say about only the sensitivity is around 70%. But when it's in the PCR and the molecular diagnostic methods, but when it's come to even the serological test, this sensitivity even goes down about 30%. So here it's in worldwide, according to the usual data, it's about like false negative results are false positive results are very less. So in case of, it's about 5% false positive results, but false negative results can be there around 30%.
Okay, thank you, sir. There's another question from Amani. There are patients who are diagnosed with COVID-19 with their first PCR test result, but the second test done prior was tested as negative. Why is that?
Yeah, before answering that, I think Nuska's question has another, but she had asked how are they verified? The verification, if you look at the verification, now better to repeat the test. If you can repeat the test, then you can increase the sensitivity and specificity further. If you can repeat the test. So usually now if a patient has almost all the symptoms of COVID-19, and if it's clinically highly suggested that that person is having COVID-19 from a clinician, but still if that person is getting negative results, then it is better to go for a repeated test maybe after 2 days, because there can be several reasons, like maybe the sample is not detect, the RNA may not have extracted properly, there might have been inhibitors. So because of that, it is always better to repeat if the symptoms are highly suggesting with COVID-19 COVID-19 symptoms. And Amani, for your question, yes, there are patients who were diagnosed with COVID-19. Okay. Now, the thing is, it can be due to indeterminate results, because sometimes when you get only one region is positive, we have to treat it as indeterminate and we need to repeat the results. But I don't know, maybe sometimes due to the reporting errors or something like that, it might have happened. So usually the advice is, that is why we use 2 regions. So if only one region is positive, we have to consider it as indeterminate, not as positive. So we have to repeat the result. But sometimes if they haven't done that, if they haven't reported it as indeterminate, then it can give false positive results. So the best thing is, when you get the indeterminate results, to repeat the test.
Okay, thank you. Dr. Donald, would you like to add some comments on the questions?
So regarding the testing, it's not an area that I'm familiar with, so I was learning from China as much as everybody else. So nothing to add on the testing questions, and I am not an immunologist, so everything Peter said seemed good to me. I have nothing to add on that either. There was a question earlier on about what's the role of a molecular epidemiologist, and this is kind of, as I said in the text, a new breed of epidemiologists who use molecular techniques. And in fact, epidemiologists, not all of them, but a large number of the new ones, will have molecular training. And one of the other questions was about what is the training required to become an epidemiologist. And I would say a basic microbiology degree, undergraduate degree, it will get you entrance, and then you would need to do a further postgraduate degree specializing in epidemiology. But the basic microbiology will give you an option to go in that direction.
Right. Thank you so much, Dr. Donald. So any more questions?
So there
Okay.
is one more question, I think, Kokol. When we talk of sensitivity, is it analytical sensitivity or clinical sensitivity? Actually, it is clinical sensitivity. Analytical sensitivity is actually very high. It's about 95% than analytical sensitivity. But the clinical sensitivity is 70%. So that means, you know, it's not the problem with the test. It's you most of the times it can be the other factors like, you know, sample collection errors, maybe the degradation of RNA when you transfer the samples. You know, if you don't use the proper methods, then again it can reduce the amount of RNA. So the clinical sensitivity is about 70%, but the analytical sensitivity is very high. It's about 95% analytical sensitivity.
Okay, so if there are any questions, you can, I'll put up an email address. You can post your questions onto that email address as well. So we would wrap up for today, and I would like to convey my thanks to Dr. Donald and Professor Balo and Mr. Chanatharanga for joining us and sharing your research experiences and your knowledge with all of us today. And a big shout out to all the participants for joining in, and hope to see you soon with another session like this. And thank you and have a pleasant evening.